Voluntary contraction of skeletal muscles can be briefly interrupted after strong stimulation of a cutaneous nerve. This is termed the cutaneous silent period. Given its onset latency, it has been concluded that the efferent arc depends on A delta fibers, while the efferent arc is an alpha motor neuron response. Although it is believed that the CSP is mediated by segmental spinal mechanisms, the origin of this inhibitory reflex is still a matter of debate. Motor neuron diseases are a group of neurodegenerative disorders characterized by dysfunction of either or both upper and lower motor neurons. In this group of diseases, loss of inhibition at the cortical level is a well-studied phenomenon. In amyotrophic lateral sclerosis, the CSP has scarcely been studied. The main goal of our study was to investigate CSP latencies and duration in different phenotypes of motor neuron diseases categorized by the severity of the upper motor neuron signs. Recordings were made on the strongest ADM muscle with surface electrodes, while sensory orthodromic electrical stimulation was performed with ring electrodes on the fifth finger at 15 times the sensory threshold. In the baseline condition, subjects were asked to maintain a stable contraction of the ADM muscle of around 50% of maximum force. In the test condition, patients were asked to maintain a stable contraction of the ADM muscle of around 50% of maximum force while doing a maximum contraction of the contralateral ADM. Auditory and visual feedback was provided to help subjects to maintain a stable contraction of the target muscle. The CSP onset latency was defined at a drop of the EMG trace below 80% of the baseline preceding the stimulus, while duration was calculated from the onset latency to the return of the EMG signal above 80% of baseline. Given the variable upper motor neuron involvement in motor neuron disease patients, subjects were divided into three groups: ALS without spasticity, progressive muscle atrophy, and a group merging patients with primary lateral sclerosis and ALS patients with spasticity in lower and/or upper limbs. We also included a group of healthy controls. The amount of EMG signal during maximum contraction, as well as the degree of contraction during the CSP measurements, was similar in the different groups for the different experimental conditions. In the baseline condition, all motor neuron disease groups showed delayed onset latencies when compared to healthy controls. There was no significant differences in the CSP duration among groups. Despite the trend for less EMG suppression during the CSP in all motor neuron disease groups, statistical significance was only achieved in the PLS spastic ALS group. During simultaneous contralateral ADM contraction, no change was observed in the control group. However, onset latency shortened in the PMA group. In this experiment, the amount of EMG suppression tended to further decrease in all motor neuron disease groups, while staying the same in the healthy subject group when compared to the values observed at baseline. Our results in the baseline condition show that onset latency of the CSP is significantly prolonged in patients with motor neuron disease independently of the disease phenotype when compared with healthy controls. When the cortical spinal tract lesion is subclinical, like in the PMA patients, activation of the ipsilateral cortex, probably due to reduced transcallosal inhibition, can compensate, causing shortening of the CSP onset latency. Suppression of the EMG signal during CSP is decreased in patients with spasticity, indicating that marked upper motor neuron dysfunction can not only slow but also reduce the amount of inhibition in CSP, suggesting that the cortical spinal tract is important in segmental spinal cord inhibition. This simple neurophysiological test could be a valuable tool in the assessment of subclinical upper motor neuron involvement in motor neuron disease patients.